Welcome back to Chapter 12. This week we are talking about units of heredity, chromosomes, and inheritance. Our first topic for Chapter 12, Section 1, is X-linked inheritance in humans. Now, certain human conditions, such as red-green color blindness and hemophilia, are called X-linked conditions. And the reason that they are called X-linked conditions is because these alleles, or the gene location where this occurs, is on the X chromosome. So if you look over here in the picture, we have our Y chromosome, we have our X chromosome, and if there is a problem at when these chromosomes get duplicated and made, if there's a problem at these specific locations, these different diseases occur. So one of those is, there's two different types of hemophilia, and there's also the color blindness. So those occur on the X chromosome. Men are more likely than women to suffer from X-linked conditions because men only have a single X chromosome. And let's look at why that happens. So in your parents, let's say you have a mother who has a normal chromosome for normal vision and a chromosome for that would lead to color blindness. Now, the normal vision allele here is dominant, and so she comes out with normal vision, but she is carrying that color blindness gene. The male has one copy of a normal vision gene on this chromosome, on his X chromosome. There is not a location for that gene on the Y chromosome. It's not as big as the X chromosome, and it does not have all the genes that the X chromosome has. So he only has one copy of that X chromosome gene. In his case, he has a normal copy, so he has normal vision. Now, their possible offspring, they can get either a normal or a color blindness allele from mom, and then they can get either a normal color blindness allele from dad or a Y chromosome that does not have anything to do with color blindness. So in this offspring, they got a normal chromosome from mom, X, a normal X chromosome from dad. That leads to a female because she has XX, two X chromosomes, with normal vision. This second female got a normal X chromosome from dad, a color blindness allele chromosome from mom. So she has normal vision, but she is a carrier for color blindness, but she is not colorblind herself. Another possibility is that they will have a boy, and the boy could have he could get a normal X chromosome from mom and a Y chromosome from dad. Since he has a normal X chromosome, he has normal vision. And our last possibility of children from this couple is that they could have the colorblind X chromosome from mom and a Y chromosome from dad that does not have anything to do with color blindness. Since they only have one copy of this chromosome, it's the copy that shows up. And so this male will be colorblind. So when a couple undergoes something called genetic counseling, this is what a genetic counselor is doing, is looking back through their family history of traits and which relatives have those traits to try to determine what traits the parents might be carriers for and what probability it is that their offspring might have certain genetic conditions. Probably wouldn't go under genetic counseling for color blindness, maybe more for serious diseases, but this is how that process would work. So another example of an X-linked trait is hemophilia, and this is where your blood doesn't clot normally because it lacks sufficient blood clotting proteins. So if you have hemophilia, you might bleed for a longer time than normal after an injury than you would if your blood clotted normally. So sometimes people with this, especially a long time ago, would die more easily from injuries that would be minor to someone else because their blood can't clot. So a woman can carry this dysfunctional blood clotting allele on one of her X chromosomes and normally won't show up with hemophilia because she has a normal blood clotting allele on her other X chromosome. So she has one X chromosome that's the hemophilia chromosome and one normal blood clotting chromosome. The normal blood clotting chromosome is dominant, and so she would not have hemophilia. She she is a carrier though for hemophilia. So just like the color blindness, she can pass on hemophilia, and it's more likely to show up in her sons than it would be to show up in her daughters. Hemophilia and red-green color blindness are examples of recessive genetic conditions we talked about, meaning the conditions will not exist if you only have one dysfunctional allele and one functional allele. You have to have two copies in a female of that allele to show up, but only if it's X-linked, you only need one copy of it in a male to show up. This is a picture here of a red-green colorblind test that a doctor might use to check your vision for color blindness, and with normal vision, you would probably see the number 8 here, which is red, and then the dots around it are green, but if you had red-green color blindness, your eyes would not distinguish between these two sets of dots, and you could tell that you have red-green color blindness. We mentioned this term on a previous slide, but just to reiterate this, a person who carries the gene, but the gene doesn't show up, is known as a carrier. So in our example, the mother who has the trait, say for red-green color blindness, that has one copy of that allele, but the other copy is normal. Her vision is normal, but she carries the color blindness gene, and she is known as a carrier. So we've just talked about X-linked genetic disorders. Now we're going to talk about autosomal genetic disorders, and these are genetic disorders that occur on one of the other 22 chromosomes. Sickle cell anemia is an example of an autosomal recessive disorder. It's autosomal because the genetic defect that brings it about does not involve the X or Y chromosome. And sickle cell anemia is an inherited form of anemia, which is where your blood cells can't carry adequate oxygen throughout the body. And in sickle cell anemia, the red blood cells become rigid and sticky and are shaped like sickles or this crescent moon shape. So here would be a normal red blood cell, nice and round, able to carry lots of oxygen. These sickle cell blood cells cannot carry the normal amount of oxygen and can lead to anemia. It's recessive because the person must be homozygous for the sickle cell allele to suffer from it. So you need to have two copies. Homozygous means two of the same copy, and you need to have two copies of that sickle cell allele in order to come out with sickle cell anemia. So you can be a carrier. Mom and dad can both have normal blood, but can be carriers for sickle cell anemia. So if dad is a carrier and mom is a carrier, then you can get one of your children who actually will end up with sickle cell anemia. Two of the children would be carriers, 2 out of every 4 would be carriers for it, and 1 would be completely normal. Some genetic disorders are referred to as dominant disorders, meaning those in which a single allele can bring about the condition regardless of whether a person also has a normal allele. So in recessive, you have to have 2 copies of the allele for it to show up, unless it's in the X and Y chromosome like we discussed. But in autosomal genetic disorders, if you have a dominant disorder or disease, you just need 1 copy of that allele to have the disease, for that disease to show up. Here's an example of looking at 2 different Punnett squares for a recessive disorder and a dominant disorder. So as we talked about earlier, our sickle cell disorder, the mother is not sick but is a carrier for sickle cell, so she has 1 sickle cell gene here, little s. Father also is a carrier, but does not show up with sickle cell anemia. He does not have the disease, but he is a carrier, another small s. So in this column here, the children will get the large S from mom. In this column here, they'll get the small s from mom. In this column going across, they'll get the large S from dad, and going across here, the small s from dad. So the only child out of these 4 that actually shows up with sickle cell anemia is this one down here, who gets a little s from mom and a little s from dad. So there's a 25% probability that a child will get sickle cell anemia in a recessive disorder when each parent is a carrier. Now, Huntington's disease is a dominant disorder, and if the mother has two normal genes, she does not have Huntington's disease, and the father has a Huntington's disease allele and a normal allele, then the children, two of them will come out with the disease because they got the large H from dad, and two of them got a small h from both mom and dad and will not get the disease. So in this case, where the mother does not have Huntington's disease but the father does, and it's a dominant disorder, then 50% of their children would have that, 50% of them would have the probability of inheriting the disorder. Okay, next up, let's look at tracking traits with pedigree. So in tracking heritage diseases, scientists often find it helpful to construct medical pedigrees, which are genetic familial histories, so looking at the genetics within a family, that normally take the place of diagrams. And pedigrees allow experts to make deductions about the genetic makeup of several generations of family members. Let's look at first just the shape of a pedigree and what we can tell in terms of the family tree from the pedigree. So when you see two people above and people below, that means we have parents above and offspring below. A line between the two parents means they had offspring together, and a line going down shows that we are leading to those offspring. So these are the parents, line between them means they had offspring together, line going down means that everyone off that line below is their child, directly off that line. So these two parents here had 3 children, 1, 2, and 3. This third child got married to this, or had offspring with this person here. So these two people here, line between them means they had offspring, and the line going down means these are their offspring. So they had 4 offspring, 1, 2, 3, and 4. This person here, if you go up above, these are their parents. And if you go over from them on the same row, this is their brother here. So, and again, when we're looking at the pedigree, a circle is a female and a square is a male. Now, if we want to put traits into our pedigree, a person who's completely colored in means that they have that trait. So blue would mean they show up with the trait. And let's say we're looking at whether the person is albino. If this square is, or circle is blue, that means they are albino. Halfway filled in for a female or male means that they are a carrier, so they're not expressing the trait, they're just carrying the gene. And then solid white means that they're not a carrier and they do not have that trait. So they're not albino and they're not carrying the gene for being albino. Now, the way a pedigree can be useful is, let's say that we have these two parents here and they go in for genetic counseling because they have a child who is albino. And those people want to know, what are our chances of having another child that's albino? And from looking at that this is a recessive trait and it appears equally on the male and in the males and the females, means it's autosomal and not recessive or not X-linked. And so the genetic counselor can look at this and tell them what their probability would be of having children who are the rest of their children being albino or not albino. So if we're looking at a recessive trait and we were to do a Punnett square for these two people, you could find out what the probability is of having a child who is also albino or not albino. Now, pedigrees can be helpful in not only determining, sometimes we don't know if it's dominant or recessive, and so by looking through a pedigree, that can help a geneticist figure out whether, or a genetic counselor figure out whether that trait is dominant or recessive or sex-linked, and all of these factors can go into helping a parent know what their probability is of having children with certain diseases. Next, we're going to look at something called polyploidy, and this is in section 12. So polyploidy is normally human beings and many other species have diploid, as we've talked about, or paired sets of chromosomes. So you have one chromosome from mom and one from dad. In human beings, this means that you have 46 chromosomes in all, 22 pairs of autosomes, and then either an XX chromosome pair for females or an XY chromosome pair for males. The state of having more than two sets of chromosomes is called polyploidy. So normally you have one set of all your chromosomes from mom and one set of all your chromosomes from dad. But if you were to get two sets of all your chromosomes from mom for every single chromosome, or two sets of all your chromosomes from dad, that would be considered polyploidy. Many plants are polyploid, but the condition is inevitably fatal for humans. So if egg cells or sperm cells were to undergo meiosis and the sperm were to fertilize the egg, and either the sperm or the egg were polyploid, then that egg would not develop any further. So it would not develop into a baby. Now, you can have an incorrect chromosome number of just one of your chromosomes, and this is called aneuploidy. Aneuploidy is a condition in which an organism has either more or fewer chromosomes than normally exist in the full set. So not a full copy of every single chromosome. Remember, you have 23 pairs of chromosomes, but just an extra copy of one of those chromosomes. And aneuploidy is, in most cases, it's going to be fatal. It's responsible for a large portion of the miscarriages that occur in human pregnancy. So when a pregnancy ends in a miscarriage, oftentimes, not always, but oftentimes that is due to aneuploidy. So the cell did not undergo meiosis properly, and the correct number of chromosomes are not present. So a small proportion of embryos will survive aneuploidy, and the children who result from these embryos are born with conditions such as Down syndrome. So there's some sort of syndrome that will come out of being aneuploid. So for example, in Down syndrome, that means that the child has 3 copies of chromosome 21 instead of 2 copies. Instead of the one from mom and one from dad, they have 1 extra copy. They call this trisomy 21. Tri as in 3 and 21 because you have that's on chromosome 21. So trisomy 21 means you have an extra copy of chromosome 21, and that leads to Down syndrome. Now, the cause of aneuploidy is usually non-disjunction, in which the 2 chromosomes or sister chromatids fail to separate correctly in meiosis. And so normally, if you remember in meiosis, and this occurs twice, meiosis 1 and meiosis 2, but the chromosomes line up at the center plate, and then they should separate, getting one sister chromatid over here and one sister chromatid over here. Now, occasionally, this separation will occur properly in most of the chromosomes, but in one of the chromosomes, both copies might go to one side instead of one going to one side and one go to the other. So if this were to happen, for example, with that chromosome 21, you would get Down syndrome. So this egg, if it was to get fertilized, would end up with, when it splits, would end up with a child with Down syndrome. This side over here that does not have a chromosome 21 at all would end up with a miscarriage. So that would not be a successful embryo that would survive fertilization. So far we've been talking about aneuploidy from meiosis, so it will happen to the sperm and eggs that occur as a result of that, but aneuploidy can also occur in regular cell division of your other body cells in mitosis. They found that a number of cancers are caused by aneuploidy. So when this mitotic division does not occur properly and you end up with aneuploidy in your regular body cells, that can lead to the development of cancer. And so recent evidence indicates that at the least, aneuploidy appears prior to the initiation of some forms of cancer. Could be possible in all forms of cancer, but they know it's at least in some forms of cancer that aneuploidy is the cause of that. Our last section in this chapter is 12.6, structural aberrations in chromosomes. And harmful aberrations or changes can occur within chromosomes, and many of these aberrations come about because of mistakes in chromosomal interaction. Some different types of problems that can occur when your DNA gets copied, and the DNA does not get copied properly. Some different examples of things that can happen are a piece of your DNA or a piece of that chromosome can be deleted entirely. So you might have this segment that just doesn't get copied over. So the chromosome ends up like this. You could also have an inversion where the DNA should be in this order here, and a piece of that DNA gets flipped over. And so your DNA then gets, the order of that DNA gets mixed up. You can have a translocation where one piece of DNA from one chromosome gets copied onto a different chromosome, or you can have a duplication where you get a section of DNA that gets copied twice, and you have two sections of the same piece of DNA, whereas you should have only had one piece of that DNA. Now, where this change occurs, on what section of the DNA and how much DNA this occurs to, will depend on whether this change is will the cell be able to just continue as normal, or whether that particular cell will just die. For example, if you were to delete a section that was telling the DNA to stop duplicating, and you deleted a stop section of the DNA would continue to replicate over and over and over again without stopping or without ever being told to stop. And that cell would just keep duplicating and duplicating and duplicating unregulated, and that can lead to the cell becoming cancerous. So the cell just doesn't stop growing. And so you end up with a cancerous growth due to that deletion. So really how harmful a change in your DNA is depends on exactly where that change is and how big of a section of DNA is impacted. That's the end of our chapter 12 lecture. Next week we'll be covering chapter 13, and let me know if you have any questions as you go about your week.